In this lesson, we'll talk about how primary antibody detection works, and we'll also examine the three main types of primary antibody that you can choose from. A target protein can also be referred to as an antigen. Primary antibodies work by recognizing and binding to the antigen by way of a region on the antibody molecule known as the antigen binding site, or ABS. Particular sites on the antigen, known as epitopes, are the specific targets that antibodies recognize. When this recognition occurs, the antibody will bind to the specific target, forming the antibody-antigen complex. As discussed in the blocking course, primary antibodies are sometimes labelled, typically either with an enzyme or a fluorophore, which allows for direct detection. But much more commonly, unlabelled primary antibodies are used instead. These can subsequently be detected using labelled secondary antibodies, and this process allows for a more universal detection, in that one type of secondary antibody can be used for detecting many different primary antibodies. It also serves to amplify the signal, as several labelled secondary antibody molecules will bind to a single primary antibody molecule. Because of its many advantages, and because it is the most widely used technique, in these courses we will focus on indirect detection of your targets. We discuss the main types of primary antibodies that you can choose from and provide detailed advice on the selection process in another Lambda U learning path, The Importance of Antibodies. But as a brief summary, these fall into one of three categories. The first of these categories is the monoclonal antibodies. These are produced by cultivating a single hybridomer B cell line, which produces one single antibody clone. Every molecule of an antibody that is produced in this way will bind to the same epitope on the same antigen. As a result, they are usually highly specific for their target proteins and will bind with high affinity. They are excellent for use in quantitative assays because of their high purity and consistency, and often result in low background because of their specificity. However, the downsides include the fact that they are expensive due to the time-consuming method of production, and their usage sometimes results in lower signal intensities due to the fact that they recognize only one epitope on the target rather than several. The second category is polyclonal antibodies. These are a collection of antibody molecules produced from different B cells, which are purified from the blood serum of animals immunized with the antigen. As a result of this production method, they recognize and bind to multiple epitopes on the same target protein. In general, they are less expensive and less time-consuming to produce than monoclonal antibodies, but they have the potential for increased non-specific detection and lot-to-lot variability. Despite all that, they are nevertheless commonly used for Western blotting, as they still exhibit a high affinity for the antigen, and can actually help to boost signal when compared to monoclonal antibodies. This is because multiple antibody molecules will bind to multiple epitopes on the target protein instead of just one, offering a more robust detection. The third and final category you could choose from would be recombinant antibodies. These are antibodies produced in vitro using recombinant DNA technology. The specific gene fragment that codes for the variable region of an antibody of interest is cloned into a plasmid, which is then transferred into an expression host, such as bacteria, yeast, or a mammalian cell line. Recombinant antibodies are highly specific. They lead to very low background, can be quickly produced, and can be designed and edited to best fit different needs. However, they cost more to develop or to purchase than monoclonal or polyclonal antibodies, and there are relatively few commercially available options at this time. When you have selected your primary antibodies from the various options out there, it is important to remember that you are not quite ready to use them in your research just yet. You also need to validate them for the particular conditions of your Western blot assay. For more information on how to select and validate antibodies, please refer to the Lambda U learning path on The Importance of Antibodies. Now we've discussed the importance of carefully selecting a primary antibody and the categories of antibody that you can choose from, let's move on to the protocol for the primary antibody incubation step.